Very pleased to introduce you to the next panel discussion on the future of neuroscience. So the last panel discussion, Chris talked about the relationship of genes that were coding proteins, and these proteins determine the function of the different cells within a particular type of tissue. And a lot of the discussion was about different types of tissues, such as heart or skin. Today we're going to be, or in this panel, we're going to be focusing in on a particular tissue, the nervous system, which of course includes the brain. Neuroscience is the study of the function of the nervous system, and in particular in humans, we tend to focus on the function of different brain regions and their role in mediating behavior. So in our panel today, we'll have an overview initially from Joe Coyle talking about the mutations in different genes that could underlie particular mental disorders. And then Bob Desimone will talk about the particular techniques for measuring and manipulating the activity within the nervous system of animals and potentially even in humans. And then Eve Marder will talk about computational techniques for linking between the molecular structures, the protein structures that are determined by genes, and the function, the physiological function of the circuits within the nervous system. So I'll start out introducing Joe Coyle. Joe Coyle is the director of the Laboratory for Psychiatric and Molecular Neuroscience at McLean Hospital. He's the Eben S. Draper Professor of Psychiatry at Harvard Medical School. He was formerly the chair of Department of Psychiatry at Harvard. He's a former president of the Society for Neuroscience and the American College of Neuropsychopharmacology, and is a member of the National Academy of Science. And as you can see from the program you have, there's obviously a lot of awards that all of our different speakers have received, so I won't try to itemize all of them. He's received the Lieber Award from NARSA and the Efron Award from the American College of Neuropsychopharmacology, and he's very well known for his work on schizophrenia and a range of different disorders. So thanks, Joe.
Thank you. So I want to thank Mike for inviting me to participate in the celebration of the opening of Kelshan Center. It's truly a beautiful building, but I think more importantly, you had the wisdom to provide the money to make sure that the science will be done there forever. Mike asked me to talk about the future of neuroscience, which seemed like kind of a big topic. And then he said, in 7 minutes. So I thought I'd talk about what I do, and I'm a physician trained in pediatrics and in psychiatry, but I've spent the last 40-some years of my life studying the neurobiology of brain disorders. And I should point out to you that brain disorders account for 35% of the burden of illness to society, so it's really quite substantial. And twice the cost of cancer. Now, about every 50 years, medicine in general and psychiatry in particular undergo a paradigm shift. And there we go. And 50 years ago, with the introduction of psychotropic medications, the interest in psychiatry went from the unconscious with Freud to the brain. Now with the genome revolution sweeping medicine, it's also affecting psychiatry. So you might be surprised to find out that genetics plays a very important role in the cause of psychiatric disorders. And in this slide, which was science review by Dan Geschwin, it shows on the right the heritability. That's how much genetics contributes to the risk of these disorders. Anxiety disorders, it's about 50%. Interestingly, MDD stands for major depressive disorder. It's about 35 to 40%. Schizophrenia, it's almost 80%. Tourette syndrome, 80%. And even a disorder like PTSD, a post-traumatic stress disorder, which clearly involves an environmental trigger, the heritability is 50%. So these risks, this genetic risk however, is not due to what we call Mendelian genetics, you know, brown eyes, blonde hair. It's due to complex genetics, in which multiple risk genes of modest effect interacting with environmental factors produce the phenotype. Now, let me talk a bit about, for a minute or two, on schizophrenia, the area that I do my research. This is a disorder that has its onset in the second decade of life. The symptoms include psychosis, rather striking impairments in cognition, and impairments in motivation. It's a lifelong disorder. 1% of the population is affected, 7th most costly illness to society. It accounts for 7% of the burden illness in society. So a big, big problem. Now, recently, a huge genome-wide association study was carried out to understand the genetic risk of schizophrenia. This involved 113,000 controls and almost 40,000 patients with schizophrenia, in which the entire genome was sequenced. And on the slide is shown at the x-axis is the entire genome broken down by chromosome, and on the y-axis is sites on the chromosome that achieve significance of association with the risk for schizophrenia. And the threshold is very high. It's 5 times 10 to the minus 8 to achieve significance because of all the multiple comparisons. And what you see, the green lines, we call this a Manhattan plot because it looks like the skyline of Manhattan. Those are the ones that achieve significance. It turns out 108 sites in the human genome are associated with the risk for schizophrenia. We've been working on one particular gene, started about 10 years ago before this study was ever done, and that is serine racemase. So this is a schematic representation of glutamatergic synapse. Glutamate is a major excitatory neurotransmitter in the brain. It drives neuronal activity, and it activates a number of receptors. One is this receptor called the NMDA receptor, which is at the epicenter of plasticity in the brain. It is responsible for laying down memories, and when persistently activated, it causes the sprouting of more excitatory synapses. Serine racemase makes D-serine, which is an obligatory coagonist at this receptor. No D-serine, NMDA receptor does not work very well. We knocked out this gene for serine racemase, no D-serine in the brain, and the brains of these mice showed the same neurochemical, structural changes we see in schizophrenia, the same cognitive impairments and impairments in motivation like you see in schizophrenia. So that's just one gene. Now, I showed you 108 sites. They're not random sites. On this slide are 8 other risk genes, and they all cluster around the glutamatergic synapse. So now we can see emerging pathways of pathology that could be targets for drug development. Now, I use the categorical diagnosis schizophrenia like it's a specific entity. Well, it turns out, as we do these gene finding studies, that some of the risk genes for schizophrenia are also risk genes for bipolar disorder. Some of the risk genes for schizophrenia are also the risk genes for autism. Some of them are for mental subnormality. So the barriers between these disorders start to get very indistinct. And when you think at a molecular level, then you get to personalized medicine about coming up with treatments directed at the etiology and not simply at symptoms. So let me talk about the future in closing. What do I see happening? Well, I see neuroscientists doing what neuroscientists do very well, and that's trying to understand how the brain works. But the difference now is that each of these proteins that they'll be talking about is linked to a gene. And there are about 20,000 genes that are expressed in the brain, and understanding what those genes do, we'll be able to link them to the risk genes that are identified in disorder. The second thing is expanding these genome-wide association studies to that whole list of psychiatric disorders, as well as other brain disorders, Alzheimer's, Parkinson's, et cetera, et cetera, et cetera, that are due to complex genetics. Now, if I proposed that 10 years ago, you would have thought I was psychotic, because to do that study on schizophrenia 10 years ago would have cost the entire gross national product. But on the, what I want to show you here on the lower panel shows you the reduction in cost in sequencing the human genome. And where it cost $100 million in 2001, it costs less than $1,000 in 2017. And so what I predict, and this is where engineering's important, engineers will come and figure out how to make it cost $10. And the other thing engineers will do is, this is an enormous amount of data. 3 billion base pairs, 150,000 people, that's big numbers. And the other thing they'll do is help us get our arms around that data to be able to analyze it. Much of our advances in understanding these disorders have come from post-mortem studies. Now, there's a problem with post-mortem studies. One is people die when they're old, not when they have the symptoms of the disorder. And two, we don't have that many brains to look at. But this is going to be transformed with the use of inducible pluripotent stem cells, where you can grow actually an individual's brain in a dish and interrogate it for its risk genes and how its synaptic function may be altered in a specific disorder, and even test drugs. Now, one of the problems with drug development in psychiatry and brain disorders is it's done primarily in mice and rats. Well, I hate to tell you that a mouse brain is not much like a human brain, and we've had a lot of failures. And so there's interest with using CRISPR technology to actually make mutations in subhuman primates who have the complicated behavior that we have to really, and social behaviors, to look at drug discovery. And the final frontier for me is the whole issue of epigenetics. And epigenetics refers to changes in the physical structure of DNA that alters gene expression without altering the base sequence. And it can be transformed from one generation to the next if it's in the germline, although it fades out in subsequent generation. And the environment is very important in impacting epigenetics. And so this is an additional layer of complication we'll have to look at, for example, with PTSD. We know that the gene that encodes proteins that respond to stress are epigenetically altered as a consequence of persistent severe stress. So I can say that neuroscience has a very full plate, and the neuroscience of the future will actually be very much engaged in the neurobiology of brain disorders. Thanks.
Thanks very much, Joe. That's very interesting work. And before I introduce the next speaker, I just wanted to wish happy birthday to Rajan Kilichan. We heard last night that it was his birthday, so happy birthday. And as they said last night, you know, it's kind of unusual that you're giving the gift to us, but we really appreciate it. It's a very nice birthday gift, so thank you. You're
going to make a gene which will make me have another 67 years.
That's it. Yeah, that's it. That's our gift. All right, so next I'm going to introduce Bob Desimone. Bob is the director of the McGovern Institute for Brain Research and the Doris and Don Berkey Professor of Neuroscience in the Department of Brain and Cognitive Sciences at the Massachusetts Institute of Technology. Bob is a member of the National Academy of Sciences and received the Troland Research Award from the National Academy, among the other awards that you see listed in the program. And he's very well known for his work on the mechanisms of attention. So, Bob.
Thank you. I'm really delighted to be here. I think the Boston science and engineering community is the most interactive, the most collegial in the world. And I'm forever grateful to Bob Brown for recruiting me into this community when he was at MIT 13 years ago. And I can tell you, the entire community is excited about what's happening here at BU, which is now made possible with the new program and the new building that has been developed on the basis of the gift from Rajan Kilachan, who I had the pleasure of meeting last night, and very happy to be here on his birthday. This is just a great, great time for BU and the city, and all of us, your colleagues. So Joe Coyle really nicely laid out the challenge in going from a gene mutation to its manifestation as a psychiatric or neurological disorder in a human patient. And so what I'm here to do is just to tell you a bit about some of the new tools and discoveries coming out of basic science that we believe will be able to help bridge that gap in the future. So you can see on the left some of the beautiful ink drawings of the 19th century anatomists who described the nervous system in different organisms. But 100 years later, what you really want is a Google map of the brain, right? You want to know like the neural equivalents of where are all the gas stations, the convenience stores, where do I get buy a shirt, and so on. You want to know all the parts, okay? And that's one of the things that the tools of genetics are giving us, as this ability to identify all the parts at a molecular level. Now, it's a tremendous amount of information, and what those studies are showing us is you have to combine this empirical approach with collecting all this molecular data with more computational approaches to understand what the data mean. And we're just beginning to see the glimmerings of that part list in just the last year. And I think this is something that's going to be developing over the next several years. So we're getting the parts list, but of course then how are they connected? And this is something that has just been coming out of basic science studies in the last couple of years, an ability to make the brain transparent. Actually, you'd see right through it and label some of the connections. And some new techniques developed by Ed Boyd in MIT allow us to blow those little pieces of brain tissue up and make a lot of these very fine scale connections visible. Now, one limitation of these methods is that, and this, what you're seeing here now is a method applied to a fly brain by Jerry Rubin's lab at HHMI, is that it's limited to small bits of tissue. And can't even do this on larger animal brains, let alone a human brain. But I think, again, the challenge over the next 20 years is going to be to develop these methods to apply to human brains so we get the Google map of the human brain, I think, which is a realistic goal in the field. So we'll have the Google map, but of course, what you need to do is you need to understand how all is functioning, how all these parts are working together. We need tools for imaging the activity of all these neurons in the Google map. And this is the techniques for doing this using 2-photon imaging with calcium transients is being now widely developed. There are people here at BU who are some of the leaders in this field. You have Steve Ramirez, who was hired here recently, Mike Hasselmo, many others, Shui Han, who are using 2-photon imaging to now start to give us a picture of how these parts are working together. Although right now, just like I told you that for the anatomy, we only do this in little pieces, for the calcium imaging, it's also now relatively limited in its field. But this is, I think, the new frontier. It's going to be expanding the activity maps to take into account the activity of millions of neurons. And that's going on in some of the work here. And then we need to be able to take those data and work with people with computational tools that are going to give us a chance of trying to understand how it's all working together. And okay, so now we have, moving through the future, we have our Google Maps, we have some of our activity maps, we'll develop hypotheses about how it's working. And one of the ways we have of testing our hypotheses is by manipulating the neurons in these maps. And for that, we have a revolutionary advance for manipulating neural activity with light. It's been called optogenetics. And you have one of the pioneers of optogenetics is on your faculty here, Shui Han, who I have to, very happy to say, just got tenure here at BU. She and She'll be speaking this afternoon. She and Ed Boyden, they discovered the method for suppressing neural activity for light together. And now this has been tremendously advanced, and we have different colored lights manipulating different neurons and so on. And this is going to be, this is an incredibly important tool. Again, you have many people here at BU, including Mike Hasselmo and Shuhan and others, who are using this to study circuit function. And finally, I've been talking a lot about work that's basically animal work, but what we want to have is an understanding of what's going on in the human brain. And for that, our main tool has been functional magnetic resonance imaging, which is, again, you have a faculty here at BU who are some of the leaders in the field, people like Chantal Stern, David Summers, and this is telling us a tremendous amount about the organization of the human brain. But I think that the challenge now in the next few years will be to improving those methods, giving us, again, we want higher spatial resolution, we want higher temporal resolution. I know you have David Boaz here now at BU who's developing infrared methods for looking at brain activity, and there's a lot of promise in those as well. One of the things that we've been doing at MIT is combining fMRI with magnetoencephalography to give us the spatial resolution of fMRI with some temporal resolution from MEG. You're looking at a brain scan with that sort of temporal activity superimposed on it there as well. And one of the things that I think we'll see in the upcoming years is what's called atomic MEG, which has the ability of getting some of these MEG maps with sensors that work at room temperature. So I think there's tremendous things going on. There's tremendous promise for what we will see in the future. And now Eve Marder maybe can tell us more about how we're going to understand all these data. Thank you.
Great, thanks Bob for that overview of the techniques for measuring and manipulating activity. So Bob kind of already mentioned this problem, and it's really central in neuroscience, linking between the proteins that are expressed from different genes and then these large-scale effects that you see, in the neural activity, of course, in the brain, looking at with fMRI, but even just looking at the physiological activity, the changes in the time course, the electrical potentials within individual neurons or interacting groups of neurons is a tremendously complex problem. And this is really where Eve's work, this is one aspect of her work. She does computational modeling of the dynamics of circuits as well as neurophysiology to understand these interactions. So Eve Marder is the Victor and Gwendolyn Beinfield Professor of Neuroscience at Brandeis University. She's also a past president of the Society for Neuroscience, and again has a long list of awards she's received. Some of the most prominent are the Gruber Award and the Kavli Award. And as mentioned, she's very well known for both the neurophysiological work and computational modeling, linking these protein components to the physiological dynamics of the electrical potentials of neurons. So, Eve.
So it's a great pleasure to be here, and I would just like to say that I feel like I'm going to introduce a bit of a word about some of the real deep problems that face us as we try and understand neuroscience. There have been this extraordinary explosion of tools of all kinds, and you've been hearing about them all morning, and yet, in my mind, the big problem that still bedevils of us, and that we actually have to use a tool that we all have, which is our own brains, is how to account for the behavior of a system in terms of the behavior of its underlying components. And I started out as a biologist, and it's now probably almost, I don't know how many, 30 years ago that I started working with people like Nancy Capel here at BU, because I wanted to understand how you could go from understanding the properties of single components to understanding the properties of a system. And basically, as we're getting more and more understanding of all the different kinds of membrane proteins that are important for electrical signaling, as we're getting more and more understanding of the genetic basis of mental disorders, etc., we still need to be able to reason from the properties of the single molecules to the cells, to the circuits, and actually understand and try and make sure that you actually have the right understanding. And I'm going to show you some examples of why you might not always have the right understanding. And I would just also like to say before going further, is I was trained as a biologist, and my lab now is almost always half computational scientists who come out of physics and computer science and math, and the other half biologists. And I think it works, this came up in the last symposium, it works much better when those people are sitting desk next to desk next to desk. Okay, now I'm just going to go back to basics to make my point. All neurons have many different kinds of ion channels. These are the proteins we're talking about. The NMDA receptor that Bob was talking about is one of them. And we want to understand how the behavior of single neurons and then of circuits depends on the properties of those channels. And there are lots of methods that we know to characterize the properties of these membrane proteins one by one. But the only way we really can assess them in terms of their overall interaction is to put them back either into a computational model or to manipulate them using some of these new tools and look at physiology. Now, I'd just like to show you a couple of the worries that I have, because I'm a worrier. Anyway, so this is a computational model. It was built many years ago in my lab in collaboration with Larry Abbott. And this is a model that has a number of those different kinds of voltage and time-dependent currents. And up there on the left, in what's A, you can see two model neurons that have basically the same properties, but they have very different sets of underlying numbers of channels. So this tells you that you can have two neurons with very different sets of channels and get very similar behavior. And the opposite can hold. Two neurons with very similar sets of channels can produce very different behaviors. So this now becomes the reason why you have to be able to build models, but it becomes a cautionary tale about reasoning too facilely from many of the new approaches that we're getting. Now, one of the things that's happened in the last few years is as computers have gotten faster and faster and bigger, it's possible to do larger and larger and larger simulations or larger and larger explorations of parameter space. So I had a postdoc even now, almost 13 years ago, who built 20 million model networks to try and find the set of parameters or the sets of parameters that would produce the behavior she was looking at. And here's two circuits. They're producing virtually identical behavior. But when you look in their underlying parameters are entirely different. So that tells you that in your brains, even though all of you have perfectly good brains, you all have perfectly good respiratory systems, but the likelihood that they are in any way very similar is very, very small. There are multiple solutions that even small groups of neurons can do to produce now that's really good. That means we're robust as a species. It means that there's a lot of diversity in the population, but this goes to the importance of the kind of individual analyses that we were talking about before, because if you want to understand brain 1, it may or may not show the same set of solutions to producing behavior 1 as brain 2. And so we are probably much more different than you might think. We're both old. Okay. Now I'd just like to make one more point along these lines. And this is now a simulation that Gabrielle Gutierrez did in a 5-cell network. And what she was interested in, I don't know how many of you can see it, she was interested in the behavior of the neuron called the HN neuron. And this shows a cautionary tale to using these wonderful optogenetic methods. In one set of parameters, the HN neuron is firing with the slow neurons. And then she did 3 different manipulations, like you could do with optogenetics. In one, she changed the properties of one synapse, and then a different synapse, and a different synapse. Manipulating all these 3 different synapses produced a change in behavior that looked exactly the same. So that means just because you find a manipulation that produces a change doesn't allow you to say, I now know the change that is responsible for this, or the process that's responsible for this, or the gene that's responsible for this behavior. Now, this kind of degenerate behavior you can see in a 5-cell circuit. Imagine in the billions of neurons in our brains. There are all these parallel pathways. And I think the real key challenge for us for the future is really understanding how to reason with parallel pathways and try and allocate information flow in a highly complex nervous system. Now, if I have one more minute, I just would like to say the other really important big challenge for the future sort of came up in the last symposium, which is that you have all these neurons, they have all these different ion channels, synaptic proteins that are important for cell signaling. Well, neurons live forever, right? Your neurons are all going to live 100 years. Right? However, and this is really important, the ion channels and the proteins that are important for cell signaling are turning over really quickly. They turn over in minutes and hours and days and weeks. They don't last for years. So that means the nervous system is constantly rebuilding itself. And so the nervous system, and this is where so many people are really excited about, but it requires a lot of really, really new tools that we haven't even thought about yet. You have to understand how a nervous system, this is a really important control problem, right? So control engineers want to think about this. The nervous system has to constantly rebuild itself to allow for plasticity, allow for experience to shape it, while at the same time maintaining robustness and stability. When you learn how to recognize a tree, you don't want to forget that when you learn how to recognize a car. And so that is one of the really big challenges that we have to understand. All these new techniques allow us, give us the parts list, but at the end of the day, we have to really understand this is a problem in control. You know, how you have loops within loops and loops, this is an engineering problem with biological molecules. And I'll stop there.
Thanks very much. Well, thank you very much, Eve, and thank you, Bob and Joe, for laying out the opportunities within neuroscience. I mean, they're challenges, but they're also opportunities for, you know, scientists love to have problems to work on, and boy, is this a big problem. And it's, you know, it's big in fascinating ways because it involves such a range of different parameters of the different channels that are in the models that Eve talked about, and these different genetic markers for schizophrenia and the different components of the circuits that Bob talked about. So it's really quite a dramatic question here, and I think it fits perfectly for the building that Rajan Kilachand helped to contribute to here and the research funds that he provided, because what they're going to do is they're going to provide people to flexibly group together from disciplines, different disciplines, to really address these difficult questions. And that's kind of what I want to start out with now. You know, Eve already mentioned that she likes to have the physicists and the engineers in her lab right there next to the biologists doing the neurophysiology. I think that's a really important thing that we have in the new building. But I'd like to talk a little bit more and elaborate on that in terms of opportunities in the future. What do you see as the next steps kind of interacting between the different data sets that you talked about and the different techniques? What do you think? All three of you could maybe comment on what you think in terms of that, and even have a dialogue perhaps about that question of what's the next step for interacting between fields to address these questions.
I think one thing that's already happening is that we're educating our neuroscience students, our biology students, very, very differently than we were 20 years ago. So because everybody understands that you're going to need computational methods to do big data analysis, to look at all different kinds of data, it's become much more important that every single experimental biologist be far more quantitatively literate than was necessary 30 years ago. So we have totally different kinds of courses now to make sure that students pick up in graduate school what they didn't learn as undergraduates, and our undergraduates often learn hybrid, so much more hybrid education.
I'd say that you know, the BRAIN initiative is doing a lot to foster the development of new technology in neuroscience. But I mean, as a practical level, I see this, how you get ideas off the ground where it might be eligible for a Brain Initiative grant. And I see at MIT all the time, our neuroscientists say, oh, there's this person over in computer science who's working on how to do recognition of cars on streets, and boy, it would really be fantastic if he could analyze how our mice are interacting and so on. And you go to them and they say, yeah, well, I don't have money to work on your problem of how mice interact or brain parts interact and so on. But we've had some donations that have allowed us to get some of those projects off the ground. And I don't know really what your plans are here at BU, but I would hope that some of those funds could be used for getting ideas off the ground, and particularly fostering the kinds of collaborations across some of the neuroscientists and engineers and people in computer science and so on that could get all new ideas and that will ultimately be funded through the Brain Initiative.
Yeah, I think the endowment from Rajan Kilachand is going to provide the funds for those types of interacting. Groups of people from BU will get together and do new proposals that bring people from the different fields. Yeah.
Oh, that's great.
So, speaking as a physician and as a psychiatrist, I think that what's going on with the Human Genome Project and the impact of genetics is so transformative. And the problem we have is we're turning out physicians, and we have turned out physicians that are really totally unprepared for this. I noted in the newspaper there was a report that only 30% of breast cancers get genotyped. When that's so extremely important in terms of making decisions about how to treat. So oncology is probably 20 years ahead of us in terms of understanding these things, but the fact is we're going to be genotyping people for what their risk factors are, and I would suggest that probably 80% of our psychiatrists are thinking back in sort of late 19th century, mid 20th century about the id, the superego, and or fairly simplistic ideas about psychopharmacology and management at that level. So I think neuroscience should have transformative effects on how we educate physicians.
Yeah, I think really it's another example. And here at Boston University, we're trying to have increased interactions between the researchers at the medical school and the researchers on the Charles River campus to really kind of impact. And I know there's already been a good interaction between the medical school and biomedical engineering, so that's starting to filter out there. I think psychiatry is in a sense one of the in a way, the wild west of research in neuroscience. It's where, in a sense, the least is known about the disorders, and the problems are the most challenging.
That's why it's so exciting. I mean, neurology is low-hanging fruit. This is where the real challenges are.
Yeah, that's right.
My comment, I think, with Joe, what you said about cancer.
Yeah.
Cancer is really out ahead. And And I mean, in many ways, cancer has provided a kind of a roadmap of how to move the field forward. I mean, there was a tremendous investment in basic cell biology that is now beginning to pay off, and that's really affecting the cancer field. And that's what's got to happen in neuroscience as well.
So the war on cancer, led by Nixon, was actually underwriting 50 years of cell biology, and now we get it. And not to trivialize it, cancer is sort of one clonal cell line that mutates along the way. We got, 20 billion neurons we're dealing with in the brain, so it is more than a couple of log units more complicated.
Yeah, so another thing I'm very excited about are some of the new techniques that Bob mentioned. He showed us a movie showing the activity being measured with calcium imaging, and we've been doing that already in the new building. We've got the sensors for doing imaging of the calcium levels in individual neurons. But I'd kind of like to talk about the future of that. For me, it's a very exciting area, and I don't know if you'd like to talk a little bit about some of the advances that you see in the future.
Well, I can tell you one of the challenges in that field is right now the calcium works best at the very, very surface of the brain. And even within the cortex, which is on the surface, you can't actually, with standard 2-photon imaging, you can't see through the whole through the whole cortex, as you well know, as you know, as in your own work. But I guess there's some advances going on that I think are really holding out promise that for 3-photon imaging, getting deeper, and then using optics. I know you have a big photonics center. I assume that you have people here that are interested in how you can get optical fibers down to do imaging deep in the brain and so on. I think that's really going to be the new frontier, is using this fantastic technology, but to expand its application to more brain circuits.
Yeah, and I think that's a really important step. I'm glad you mentioned the Photonics Center, you know, because even in the work that we've done in my lab and Howard Eichenbaum's lab, in particular, Howard Eichenbaum's lab has been doing imaging of the hippocampus, which is, you know, it's about 1 millimeter down into the brain, and to actually do calcium imaging there, unfortunately, at the present time, they have to remove this millimeter of cortical tissue from above the hippocampus, which is obviously playing a normal functional role in behavior. And so it's a challenge. They have to remove that tissue and so that it can put the lens down there to image the neurons of the sort that you showed being used with calcium imaging. And this is where the 3-photon technique is very exciting. And in fact, Tom Bifano, the head of photonics, and David Boaz, the new director of the Center for Neurophotonics, have already been talking about putting together a group and have a group of engineers and physiologists that are using these 3-photon techniques that allow them to go deeper, essentially to, you know, scan and image the calcium levels, you know, up to 2 millimeters down into the brain, so you wouldn't have to remove that millimeter of tissue over the hippocampus in order to see the activity.
So, but for me, one of the big challenges and one of the most exciting things that's happened in the last number of years is the ability to increasingly go into the human brain and get recordings during surgeries, et cetera. So autopsy material obviously has its problems, but more and more and more neurosurgeons now are better and better trained and are working with people with computational methods to capture as much information as is possible in an ethical fashion from human brains in living people who can tell them what they're seeing and feeling. And to my mind, that's going to be tremendously useful going forward. It also turns out that human brain cells may be in some ways more robust and better suited for physiological investigations than rodents. The cells are bigger and they live well, and so I mean, this I think is a question of education, which is to figure out how to optimally capitalize on what one can from all the neurosurgeries that are going on all over the country, whether that means, you know, the tissue that gets taken out when you go in for a cancer resection, or whether it's the tissue that gets removed during an epilepsy surgery, or whether it's what the information you get when you're implanting a DBS electrode. There's a tremendous amount, if we were to think collectively, intelligently, about the design and analysis of those data, I think they'd be invaluable. So,
actually, electrical manipulation of brain function is very active in psychiatry. Deep brain stimulation, aside from being used to treat Parkinson's disease, is being increasingly used for intractable depression and obsessive compulsive disorder. But with these new techniques where you can engineer genes to have channels with fluorescent tags that you can activate, I don't think it's you know, unreasonable, which we're doing in animals. I don't think it's unreasonable to see that as interventions in the future as Eve works out what these circuits are really doing. You know, like epilepsy now is a neurosurgical condition. Maybe in the future.
But you know, dreaming 25 years from now, you know, now we put in a big heavy DBS electrode and you just stimulate everything. But imagine you could express something in a single cell type and go in with a fiber optic, and now you can manipulate not everything near the electrode, but just the targeted groups of cell neurons. And so that's dreaming towards the future, but you know, I would expect in 15 years people will be doing it. And especially for people who have intractable problems where we now know some of the genetic and molecular tools, and when there's some rational ability, actually people will try before there's a rational understanding, but that's something else. I mean, no one in their right mind would have ever tried DBS, right? I mean, so.
Yeah, it is kind of amazing. And
it works really well, right?
Yeah. It's amazing that, what neurosurgeons will try out, and I'm glad they did for that. But in that case, it's interesting because it does kind of suggest that it, to some extent, you can kind of explore what happens, obviously, within limits. But it's exciting that the deep brain stimulation was developed, and it's exciting the idea that people are talking about combining it with the optogenetic techniques to be able to selectively activate particular populations of neurons.
The DBS example, I think, is a good one because it was serendipitous in a way, in that actually, they thought they actually had a mechanistic understanding of the disorder, and that led to DBS. But actually, the mechanistic understanding they had was wrong.
Exactly.
And what we have in psychiatry is all of the drugs used in psychiatry are based on serendipitous findings where things worked that without having a mechanistic understanding. And I think really what we need in the next couple decades is we need the mechanistic understanding. That's what's happening in cancer. They have mechanism-based treatments, and that's what we desperately need in this field.
And I always tell my students, I find it very amusing and very scary that the same drugs are often prescribed for chronic pain, for schizophrenia, depression, seizure, etc. And they often work in all of those things in different people. So that tells you how little we fundamentally understand. But they work in some people.
Yeah. Let me make emphasize your point. Actually, the drugs we use in psychiatry, almost all of them, in terms of molecular action, were developed over 50 years ago. Antipsychotics, antidepressants, lithium, anxiolytics, 50 years ago. We've tried to rely on serendipity since then, and we've come up with a lot of dry wells. As a matter of fact, the major pharmaceutical companies have gotten out of developing drugs to treat brain disorders because there's been so many failures, and cancer is the low-hanging fruit, if you can, shocked to say that, but it is. So you're right. That's why this genetic revolution is so important, because you're starting to get at etiology and not simply symptomatic treatment.
Yeah. It really does kind of highlight the importance of developing theories that can account for the genetic data and link it to the molecular components, to the protein structures that are mediating the currents through the neurons, and then all the way up to the rhythmic activity within the basal ganglia and the cortex. And there are people here at BU, Nancy Capel and Shui Han, have been working together on, what is it deep brain stimulation would be doing? What are these manipulations doing to the network oscillatory dynamics within these circuits that could change things in Parkinson's disease or other disorders?
But I think it's just like in cancer, many tumors become resistant to chemotherapy as they evolve or whatever, and we know that that's the biggest problem in much of cancer therapeutics. Well, in the brain, we're going to have to face the fact that, as came up this morning, you have this tremendous litany of adaptive mechanisms. So you go in with a drug, and then the drug itself may produce some symptomatic relief, but it will cause all sorts of other compensatory mechanisms to be engaged. And so the brain that you see half a year later is not the same brain that you started with, and often that's to the good. And miraculously, sometimes these drugs work for really long periods of time, but sometimes they don't, and they don't because of the brain's normal adaptation mechanisms.
That's why this the stem cell strategy I mentioned of creating your own brain in a dish is so important.
Well, you're creating a piece of brain tissue with the genetics. You're not creating the brain, right?
No, I'm being facetious. All right.
Well, no, but it's really important that nobody misunderstands. So the goal would be, it's not creating your brain in a dish. You're creating a piece of brain tissue with many of the genetic and molecular properties and maybe some circuitry that resembles it, but it's not your brain. Your brain develops over 70 years as a consequence of brain.
Right, but the point I wanted to make is...
Yes. No, but this is subject to misunderstanding.
There's actually 2 points I wanted to make. One point is, for example, schizophrenia, we make a diagnosis and we think that's the same brain over 50 years. And in fact, the disorder evolves over time. So that's the first thing, because almost all the postmortem studies, that's where we get all our information about the pathology, comes from people who died. They died of old age, and they've been exposed to drugs for 50 years. Well, that may be very different from what's going on in the initial phases and the type of intervention you'd make. And the second thing is, you can get at a more individualized understanding of what is not working. Obviously, you're not dealing with a whole brain, as you pointed out, but you can start interrogating circuits that would be very difficult impossible to do currently.
You know, this issue about the evolving disease, it points out the importance of prevention. That
yes.
Certainly, you take something obvious like Alzheimer's disease, you would certainly rather to prevent it at an early stage than have to deal with a brain that's already had very significant brain damage. Well, if in schizophrenia, which is often developing in people around 20 years old, if you had markers that showed the development of the disease, and you could go in 10 years earlier and prevent it developing, I mean, then the clinical problem becomes much more tractable.
Not to get too deep in the grass on this, but most individuals who ultimately develop the psychosis, and we say they're schizophrenics, before they develop the psychosis, show cognitive impairments, are socially awkward, physically awkward, and the problem is about 7% of the population would satisfy that description. but only 1% develop schizophrenia. So preventive intervention could be very effective.
And remember, not all schizophrenia is the same, right?
Right.
Right. I mean, one of the advantages, one of the things that will come out of all the genetic work is we will be able to then subclassify, different diseases. And some of them may be very amenable to certain kinds of therapies, and others may be much more amenable to other kinds of therapies. I mean, I think with any of our major psychiatric disorders, I mean, one of the biggest hopes would be to get really, really good subclassification.
So we've already touched a little bit on, interacting at the level of the human brain, but I'd like to just mention in the context of having the new Center for Cognitive Neuroimaging in the new building, I wanted to talk a little bit about the, human imaging techniques and what you see for the future there. You showed a little bit in your talk.
Well, I've just been learning about this. We have a MEG facility, and one of the things that there's another one over at MGH. I don't know if you have plans for one here, but one of the things about MEG is this great temporal information, but it's a very cumbersome technology. And I've just been learning myself just recently about this something called atomic MEG, where these room temperature sensors that are potentially going to allow us to have MEG measurements and people moving around and not in the head in a thing of liquid helium, so I think that's a potentially really exciting development on the horizon.
Related to the deep brain stimulation, I mean, there is a move towards doing non-invasive stimulation. I'm kind of interested in what you think of those techniques, the transcranial magnetic stimulation or transcranial direct current stimulation. What do you think of those as possibilities for the future?
Yeah, just went over a review in that. We still need, there's a lot of anecdotal reports, but there hasn't been enough really carefully controlled studies. And I think obviously the technology has to be refined over time. But again, you can use them, for example, to silence speech areas of the cortex and see what's going on. Mm-hmm. So again, I think there's possibilities there that these could be much more effective techniques in the future.
Yeah, I mean, it's been an interesting experimental tool, and I also think it's something that connects to these efforts to understand the dynamics of the circuits.
Yes.
In the sense that if we really can understand the dynamics, what are the most relevant oscillatory dynamics within the cortex? These are things that can potentially be influenced by the non-invasive techniques, and and there's some evidence that they're effective, at least in depression, which makes sense in the context of the techniques that have been used for inducing seizures to treat depression, which are kind of the extreme form. But if you can do a technique that's less invasive than that, that relates to, in a sense, what we need to do is we need to take this huge parameter space of possible frequencies and possible locations for stimulation and determine what are the relevant, frequency bands that are, the ones that seem to be most relevant to the particular disorder being treated.
Absolutely.
I think we're coming to the time when we should take questions from the audience. So I don't know whether...
Got one over here. I was wondering if you could talk a little bit about the data analytics involved in this. Are you using some of the advanced machine learning and AI techniques to do your research?
People are. So there are people all over the world who are doing all kinds of advanced analytics, both for genomic data, for neurophysiological data, et cetera. But the field of computational neuroscience also has another problem, which is what we call the big Google vacuum cleaner, which is that many of our best and brightest young scientists are go off to Google and Amazon and work for them. So some of us have sort of some mixed feelings about this, but yes, people are using all kinds of new advanced machine learning methods.
Yeah, I'm glad you mentioned Google, because some of you have probably heard that DeepMind is a company that was started separately and then purchased by Google, and it's had a huge influence, partly by taking up the computer scientists. But I think it's maybe a different group of people. But it's been inspiring, I would say, for the field in getting everybody very excited about artificial intelligence. And what I'm seeing from DeepMind recently is a genuine interest in the biological circuits. They know, despite the power of their algorithms, they know they are not as powerful as the human brain. They're these feed-forward networks, like the AlexNet, that can recognize a cat in a number of different stimuli, but can't recognize, what part is the cat's tail, for example. They can't do, random different questions that a human child could do. And so really, they're looking for answers from neuroscience, both at the network level and at the computational neuroscience level, to understand better what's going on. I'm going to tell you one
very funny story. There are tons of people are using these methods, and they're clustering things and learning a tremendous amount. I had a data set in my lab that we tried any number of different kinds of tools to analyze, and finally my graduate student sat down and looked at the data and hand classified it. Because she came back and she said, and I looked at her and said, her brain is better at recognizing these patterns than any of these other methods, because she doesn't have years of data, right? In other words, the data set wasn't large enough to take advantage of some of the really big methods. And so at the end of the day, the human brain is still really good,
Eve, I remember being a grad student and reading your papers on dynamic clamp.
Yeah.
These were one of the first experiments showing that you could actually introduce active conductances and perturb the activity of neurons. So I think the field might move into actually introducing perturbations into populations of neurons and seeing how these perturbations change the variability of neural networks. I was wondering if you could comment on how that's going to play a role into understanding.
So, I think the dynamic clamp, which was done at single neurons or small numbers of neurons, has, in a way, people still do it. It's one of the methods used all over the world. But I think optogenetics has, solved or is solving some of the same problems by giving you the ability to manipulate the activity of a whole population of neurons. The limitation of classic dynamic clamp is if you have a group of 1,000 cells and they're working together, you can't manipulate them all the same way, and now optogenetics allows you to do that.
So how do you think, if you had now a method of dynamic clamp at a light level, what kind of questions would you answer?
With dynamic clamp?
At the light level.
But I think people are developing feedback optogenetic systems. So I think I've seen them. You basically just have to close the loop, right?
What would you try to answer?
Me, I personally, this is heresy, I personally would never work on a large network. So I wouldn't do that personally, but other people will. Mike will, right?
This is a layman question, so pardon its lack of technical. Dr. Marder said something that just triggered this thought. I'm wondering, just like a healthy organism fights invading viruses or whatever, is it possible that an unhealthy brain, a schizophrenic brain or whatever, will adapt and use its plasticity or whatever to fight a medication or a treatment?
It does. I mean, I think the challenge with working with every brain, healthy or unhealthy, has mechanisms that are designed to produce adaptation. If we're clever, we figure how to use those homeostatic adaptation mechanisms to produce a cure. And many medications, I think my sense is, many medications give you a little bit of an edge that allows the brain then to cure itself. So, you know, I have family members who were schizophrenic 20 years ago and they're perfectly normal now. That is to say they're living in the real world and they're working and they're you know, doing normal things, right? On the other hand, you can have those same adaptation mechanisms, homeostatic mechanisms, create disease. So these same mechanisms that can actually help produce stability can also produce pathology. And so what we have to do is understand those mechanisms better and then learn how to help the brain bias itself towards producing a cure or producing further pathology. And so that's what, you know, and the drugs are just going to be your way of sort of pushing you a little bit in one way or a little bit in the other way.
Okay, another question on the left-hand side.
Well, actually, I was just going to say on that same question, I mean, I think in some cases the adaptation mechanism, as you kind of suggested, the perturbation caused by the drugs could actually help the brain cure itself.
Right.
Like with depression, the fact that the drugs take 2 weeks to have their effect, it seems almost like the homeostatic response to the drug is helping with the disease.
Question. So there was a discussion on various computational methods. As you look ahead, what are your wishes from the computational and learning community? What do you think are the challenges in terms of new methodology development? One, I guess, potential direction is adding more biology to some of these methods that Mike already mentioned, but do you see other challenges that need to be addressed?
Well, I mean, just to jump in, I guess, if, you know, I think that in a sense, data mining is one area of opportunity for, you know, using some of the techniques from computer science and engineering and addressing this, you know, increasingly massive amount of data being generated in neuroscience, you know, both in terms of the genetic markers, but also in terms of the imaging data, even the neurophysiological data. There's such large data sets. And you know, I guess Eve said that they've tried to do that, and the human, you know, graduate student was better at it than the algorithms.
For some things, but I'd like to point out so the problem that we have now is many of the methods that exist sort of assume or require stationarity in the biological data. That is to say, you're going to train it up on some data and then use another data set, and the implicit assumption is that you have stationarity. And biological data are never stationary. And so I think trying to understand how to get away from, I mean, one guy told me that, yeah, we could analyze that, but you need, you know, 110 hours of recordings. And I'm saying, well, my nervous system is going to last 110 hours. So I think so for some kinds of data, you have very long stretches. Some kinds of data, you have small snapshots. And so trying to figure out how much information you can get from non-stationary data is really, really, really something that I'm not sure people have really thought about hard. You know, and so that's my sense. I mean, you know, there's only so much you can do if you've only got, you know, 3 minutes of recordings and then a different 3 minutes of recordings, and they're not the same.
Yeah, there's real opportunities really for development of new mathematical and statistical techniques, and you know, to address questions like having short data sets or non-stationary data sets. And network analysis, Eric Kolachik here in mathematics and statistics is dealing with developing new techniques for network analysis, because obviously the networks we're dealing with are so enormous and complex. And Uri Eden's been dealing with stochastic point processes. These are, again, new techniques that are specific to the neuroscience data that haven't really been addressed before.
From my point of view, the computational tools are being applied to the analysis of data, which is very important. But then also there's the computational problem of how to build a theory of operation, sort of how to understand what's going on in the system, right? And right now, I think we're maybe at a tipping point where just a few years ago, we weren't able to distinguish between different neuron types based on their connectivity, different cells, and so on. And so the computational theorists were faced with lumping a whole lots of neurons together that we didn't have distinguishing data. Some had connections to one thing, some had connections to the other. If you don't know that, then you try to build a theory based on a whole bunch of complex stuff that doesn't make sense necessarily. But now that we're getting that information, we can say, oh, now when you build your model, you should know that this kind of data comes from this kind of cell, this kind of data comes from this kind of cell, and so on. And I think we're going to see a new generation of models come out of that takes more into account the individual properties of the cells in the networks.
Yeah, the models are starting to incorporate some of the different subtypes of inhibitory neurons, and especially the feedback dynamics is really important to include.
Layman's question. I heard this debate about creating a brain. And
He's not going to rebuild your brain.
No, but what I understood was the fact that a brain tissue has been created obviously in the lab. So do you foresee a time where parts of a brain which are damaged in accidents could be replaced by this methodology?
I think some people would hope.
Organs?
Yeah, I mean, I think some people might hope that you'd be able to, for example, in a patient with Parkinson's disease, replace some of the missing cells. There you have an advantage that the dopamine acts sort of as a global modulator, so maybe you can get away with some of that. I think people might imagine trying to rebuild parts of the spinal cord, where again, it's sort of fairly, but I think you would always imagine, depending on the interactions between the healthy nervous system and it making the appropriate connections, so that you might be able to build a little piece of it, but then you would be able to hook it up with the healthy nervous system. Just like, I mean, I think it's really important to think about how well people hear with cochlear implants. The cochlear implant is a terribly, terribly, impoverished device, and yet people hear with them. And that's because the brain takes advantage of any information you can give it. Right? The brain learns to benefit from this very impoverished device. So I think that one might imagine, in some cases, you could give a little bit of a help, and then the rest of the nervous system would somehow or other adapt to it and take advantage of it. So I think there are all sorts of things that one could imagine.
Actually, it's taking place as we speak. Kwang Soo Kim at McLean, using stem cells grown from the fibroblast obtained from the individual, has developed techniques so he can differentiate them into dopaminergic neurons. And those have been transplanted into a patient in their substantia nigra, and hopeful that this may help repair the loss. We do know from studies done with transplanting tissue that those cells will differentiate or will grow and innervate the striatum and reduce symptoms. But the advantage of this is when you're using cells from somebody else, you can have immune responses. This is the cells from the very individual, so there's no immune issues. And again, not a brain in a dish, but you can grow dopamine neurons in a dish. You can grow GABAergic neurons in a dish. You can grow GABAergic and glutamatergic neurons from individuals in a dish and look at their circuitry. So this is in its infancy. And what's important now is before, to transform these cells, you had to use viruses. And viruses actually kind of screw up the genome and introduce potential problems like cancer, et cetera. These cells are transformed just with trophic factors. So there's no intervention that would, that we know would disrupt the genome. So it's going on as we speak.
One more question.
Like this is just the early stage for the stem cell research. We can only culture a piece of tissue in the petri dish right now.
Can't hear you. I don't know if anybody else can.
It's on. Just take it.
Okay, I know like it's still on the early stage for the stem cell research, so we can only culture a piece of brain in the petri dish now. But I can imagine that all the brains, like sort of for 60 years brain or 70 years brain, so all your memories or your experience are recorded in certain forms, like number of neural cells or the, I don't know, epigenetic level, like the level of the methylation, something. So eventually, the later day, like all this can be copied, like in the petri dish. So eventually, we probably can have a brain, like 60 years brain in the petri dish. And also, I would like to have everybody know, like now we can have a mini brain cultured in the petri dish. If you have time, you can look at the paper published on Nature by the first author is Lancaster. They can culture a mini brain, which consisted like part of the forebrain of the human. Just like the doctor said, that you can just take the cells from like the fibroblast or the blood cells and reprogram into the iPS cells, and then culture these cells into the brain tissue in petri dish. So this is still on the early stage, but in the future, I can imagine that this can be real, like with all the memories, with all the experience of the whole the human life.
That's if you could read it out.
I'm sorry.
And so that, you know, even in principle, you'd have to be able to read out all of that, and it's not obvious how you would do that. But I might just remind you of something, and that is identical twins start out with the same genetics, and their brains are not the same, right?
Right.
Even, so I think, I understand where you're going, but I just, part of me says that it's really partly a false path.
But at least in terms of diseases, you know, with the new techniques for growing three-dimensional cultures for testing Alzheimer's disease, there you're not trying to recreate the individual, but you are able to recreate the disease, and especially, you know, with something that really hasn't been showing up well in the mouse models of the tangles. And
right, I agree, you're much better off, you're absolutely much better off trying to do that with human tissue than with the rodent tissue. Absolutely.
Yeah. So there are exciting opportunities, at least for having like an in vitro preparation of a small amount of tissue with a disease state that you can then test the influence of various therapies on that disease state in the dish. I think we have to conclude there, but I really appreciate the great discussion we had. Thank you for the lively discussion, and thank you for the questions.